Okay, good morning, good afternoon, good evening. I'm Cherry Kubota. I'm a professor at the Ohio State University. Welcome to Indoor Ag Science Cafe, April. Really appreciate you come live to listen to this webinar. We have been doing many years now. This is actually a teamwork funded by a USDA program, Michigan State University, Ohio State University, Purdue University, and University of Arizona working together to advance CEA forward, specifically in the indoor soil source lighting facilities, also known as vertical farming. All right, so that's little quick announcement before starting today's presentation. So just a little schedule. So today we're gonna have Dr. Yongran Ji from Wageningen University. May we have industry speaker, and then also June. And the May one is going to be from 3M. And then this is more like a greenhouse CEA technology, sunlight alteration in terms of spectral quality. And then June is more like automation or the smart agriculture type of program. The speaker is located in Japan. So we're gonna shift the starting hours 1 hour again to 10 a.m. Eastern rather than normal 11 a.m. Eastern. So so that's the upcoming schedule of the Indoor Science Cafe. And then we usually don't have any cafe in July, and then we will start again in August for the new academic or the fiscal year. So just wanted to remind, we have a lot of lectures, basic knowledge. So if you are new in this space, I think this would help to jumpstart, particularly if you're business side and not knowing horticulture side or system side. I think this is a really good overview. So I encourage that to check out the basic knowledge of indoor farming, OptimIA University contents. All right, so today's special speaker from accessing from Wageningen University Research, Netherlands, Dr. Yongran Ji, who is going to talk about quite interesting application of indoor agriculture, indoor farming environment for breeding. So the speed breeding is a really good area if you are looking for new applications. And then he's going to talk specifically about how far it contribute to accelerating that speed breeding. So even faster than fast. So that's the topic he's going to talk about. So I'm going to have Yongran here, and then it's going to be recorded. Well, actually, we are recording, right? And we have more time for Q&A, so the use chat area, also microphone at the end, not during the presentation, but at the end, after presentation, to ask your questions. Okay, so I'm going to stop sharing and have Dr. G start presenting.
All right, thanks a lot, Cherry, for inviting me for this talk. And obviously, me being a big fan of this this Ag Cafe, and I believe this is probably my second time on this. And for a young scientist who just started reasonably young compared to a lot of the audiences, let's say, and I would say this is a great pleasure and privilege to be here. And my name is Yongran, and I work as a postdoc at this moment at the Horticulture and Product Physiology group of Wageningen University, and chaired by Professor Leo Macelis. I think he's more famous than me at this moment. And I work within a large project called Sky High, which focuses on understanding the science and technology and integration of vertical farms in modern agriculture, but also in modern society and also in modern city designs. So, and I have a small postdoc part in this project that is interestingly also partly funded by FAR, Foundation for Food and Agriculture Research. And I guess for the American audience, this is a more known name compared to the Dutch scientific organization, the NWO, which is the main funding agency of this program. Now, so today I'm going to be talking a little bit about what I've been doing in the past years on the topic. And if you have been to, let's say, if you have been to in the past year, Greensis and also Vertifarm in Bologna, then some of the slides you might have seen already. I tried to put something new here and also include a bit of new discussion so that even if you have been there, you don't end up with the same slides multiple times. And if you're going to Lansing next month, don't be surprised to see some of those slides as well. But anyway, so the whole project started with one of the urgent requests nowadays. We have vertical farms going around, we have modern controlled agriculture, but what about the varieties? We can't just assume that whatever varieties that has been selected has been bred for greenhouses for open field production would do well in vertical farms. There's different backgrounds about this. Well, first of all, the most important thing, or the most important difference between a vertical farm or let's say a more closed vertical farm compared to other CEAs like greenhouses is that we have totally different light. We have almost zero sunlight in a vertical farm. And if you compare that to different artificial lights, as you can see here, that the slides are just, the spectrums are just completely different. Now, this obviously raises the question about what do we expect, or what should we expect from a variety that is specifically bred for vertical farms, let's say. And I'm a big fan of plants, you know. There are different types of young people. Some likes planes, some likes cars, some likes guns and things. But I like planes. Now, and I always like to compare a vertical farm with this beautiful jumbo jet of KLM. They share quite some interesting similarities, and one of them is that it only makes money when they're operating, which means for a modern variety that is selected for vertical farms, they need to be able to, or at least we should try to look for possibilities for them to sustain a longer photo period, for example, or even continuous lighting. Plain sitting on the ground is not going to make profit. A vertical farm that is not operating, that is staying dark, also would make it more difficult to be profitable. And also, every cubic meter counts in a vertical farm, and every cubic, just like on the plane. So we also need varieties that can handle higher density, can have a specific architecture that is suitable for a vertical farm. And modern planes nowadays, they fly by wire, which means a lot of automation is going on. And we should also select varieties that are more automation friendly, let's say, when it comes to architecture, when it comes to certain traits and phenotypes. Now, I have a breeding background and I talk a lot about breeding, but obviously my role in this project is not actually doing those breeding. But then the background of this project comes from the urgent need of modern varieties that are specifically selected for vertical farming. We need them, and we need them fast. For many of you, I assume that you might have heard of the term speed breeding. Well, if you haven't, let's say this term becomes very popular or got popularized largely thanks to these two big papers published by Professor Lee Hickey from Queensland, I believe. And if you really think about it, speed breeding is simply the use of artificial lighting, supplementary lighting in this case, a longer photo period or tailor-made photo period, together with nutrient, irrigation, hormone application, all of those tailor-made conditions to shorten or to maximize the developmental phase or developmental processes of a certain crop. For most of the crop, that means a faster seed to seed cycle. Now, in these papers, they have given several examples. For example, here we have wheat, we have barley, we have also wild relatives of cereal crops. And they suggested that compared to, for example, a open day condition or a 12-hour neutral control, by having a certain photoperiod, a certain certain temperature, it would lead to a drastic shortening of flower or seed to seed cycle. And this is one of the graphs from those from the papers. And we don't need to go to the details here. Every different symbol marks a significant important processes during the seed to seed cycle. But the main message here is that the shortening of seed to seed cycles via using a certain tailor-made condition. And obviously here, the blue ones are the controlled treatment, while the green ones are the speed breeding approaches. Similarly, here we have also the other crops like canola and chickpea. That could be that similar treatment could be applied, and strong effects are observed. Now, these are, of course, the publication from others. And in that paper, they also reasoned that the use of controlled environment agriculture, and specifically climate chambers and vertical farms, are extremely helpful in this case. So within our project, we started this my postdoc project where we look into not only breeding for vertical farming, but also more importantly, breeding with vertical farming. Now, can we use vertical farm, and can we develop technologies and application methods to speed up the breeding process of vertical farming crops? Now, if you look at most of the vertical farming industries nowadays, or all the publications that are being produced, I would say it goes without, it will be easy to agree upon that lettuce is definitely one of, if not the most discussed and studied crop in a vertical farming background. And of course, lettuces, they come in different shapes and sizes and colors, and we eat them in different ways. But they do share, of course, some common background. They're usually long day plant, and they have reasonably long flowering times. And there are quite a bit of variation in flowering time. I pulled these data from a screening experiment from the gene bank we have here in Wageningen University. Out of the 268 genotypes that has been screened in a regular growth season, I believe it's an open field experiment. The message here is that more than 50% of those lettuces took them more than 15 weeks to flower. Now that's quite a bit of a time if you think about it. And there are some extreme varieties that could go up to 20 weeks. Of course, there are also faster varieties. The thing with lettuce nowadays, I guess, is that if you're a lettuce grower, you wouldn't want them to flower, or actually you don't want them to bolt in the first place. Well, bolting, for those who doesn't work with lettuce, bolting is the first stage of flowering. It triggers, it's marked by an extension of the stem, and then after that, the leaves becomes bitter and the texture changes, and it loses its commercial value. So in this case, if you're a grower, you would try as much as possible to prevent bolting. But the thing is, if you're a breeder, on one hand, you select your breeding varieties that are strongly selected against bolting or flowering in this case. But as a breeder, you also, it's almost like shooting your own foot. You're breeding for something that is not helping the breeding process. So there's a bit of an issue here. Now, so our question becomes, can we, without changing the genetics of the crop, can we use environmental factors to speed this up? So we took lettuce as a case study. And of course, there are things that are already well known from literatures that are up to even 50 years ago that studied what affects flowering or bolting in this case. And we know already the high temperature leads to earlier flowering, a longer photoperiod leads to earlier flowering because it's a lumpy crop. And also stresses have something to do with flowering. So, okay, we know this. Can we make it faster? And can we make it faster in vertical farms? So that was the first question that we tried to address. So it's very straightforward. We took two different lettuce varieties. One is a Batavia type, one is multi-leaf type, sponsored by our sponsor company Nunim's, part of BASF. And then we start with we call it flowering promotion condition or bolting promotion condition. These are just based on experiences that under these conditions, these two lettuces should flower much earlier than a natural growing condition, for example. So 30 degrees, 20 hours photoperiod, 300 micromoles of light, and 800 ppm of CO2. More than enough to support the growth and to trigger the bolting. So we created this condition in our vertical farm. And so 23, 24 weeks, these are the times registered by breeding companies when they grow them in their greenhouse in a normal season without actively trying to speed up or slow down. So this is basically our benchmark. And when we move them into our to our vertical farms, same genotypes, we already significantly shortened the flowering time down to 16 weeks or 18 weeks for these two genotypes. Well, great, good result, things are working, but of course these are nothing new. People know that already 50 years ago, and there's nothing new about it. So can we make it even faster? So this is where my research started. And I worked for my PhD back in the days on the far red effect on tomato. So for those who doesn't, I believe many of the audience would already know what far red does. But for those who doesn't, far red is the part of radiation that ranges between 700 and 750 nanometers. Well, technically I used to say that it's 700 to 800 nanometers, but then I was correctly corrected by Professor Bruce Buckley earlier in Italy that, hey, yes, it goes to 800, but then it's only working until 750. So far red, 700 to 750 nanometers. It's not driving photosynthesis independently, but it does help. And plant can sense it as signals, and it triggers a set of responses, for example, changing leaf architecture, changing plant architecture, changes in biomass partitioning between organs. That's actually my whole PhD. And for many crops, it also speeds up the developmental processes such as flowering. So it's one of the usual suspects, and if you ask me, then it's very natural for me to see if that works for lettuce. So we took our flowering promoting condition and we set up a very straightforward experiment. We give far red and see if there's still room for improvement, because we already shortened it quite a bit from 24 weeks to 16 weeks. Can we make it even faster? Well, the answer is obviously yes. So we managed to shorten it from 16 weeks under 30 degrees temperature to, and if we give far red on the same condition, there's still quite a bit of room to gain, and we save another 5 to 6 weeks by providing far red. Great. Now, of course, we also looked into, so we broke it down, and then we tried to figure out, okay, how many days are being spent for each of the important stages. So the vegetative phase is counted as from transplanting to the initiation of bolting. And the bolting phase is calculated as once the bolting starts until the appearance of the first flowering bud. And the next phase is between the first flowering bud and the first open little yellow flower. So these are the control conditions. So without far red, this is what it roughly takes for the multi-leaf variety to flower. And when you add far red, then we save 4 days on the bolting. We save 16 days, quite a bit, between bolting and the bud appearance, and we saved 12 days between the appearance of the first bud and the occurrence of the first flower. So the main message here is that we see the shortening on both on all the 3 stages, but what's the majority of time saving comes after bolting. Of course, if you're a breeding company, yes, you get all the variety, you get all the earlier flowering, you get earlier seeds, great, but it only makes sense if the seeds are viable, if we can use the seeds for the next round of production, next round of germination, and the next round of generation. So it's only natural for us to conduct a test to see whether germination is affected. So we took the seeds harvested from these 2 varieties, from these 2 experiments, and we stored them for 5 days in darkness at 4 degrees, and then we moved them to a germination stage of 20 degrees, and then we observe how many of those seeds germinated. And by the way, these are standard testing procedures provided by breeding companies. So, as you can see here, providing far red does not really harm or affect any of the germination rate. And they germinate. Well, all the seeds are germinated at the end, of course, and then also even after 4 days, when we move them under light, most of them already germinated. So that's great. So what we know out of these experiment, this experiment, is that for adding having a higher temperature or a better controlled environment in a vertical farm, moving them indoor already accelerates flowering, and we get earlier flowering. And after that, when we add far red to them, then we get a accelerated bolting. Well, we save 4 days there, but then the majority of time saving appears from between bolting and bud appearance, and between bud appearance and first appearance of the flowering. And we're not compromising the seed quality of this. So that's the first experiment that we did. And of course, then it's only natural for us to follow up on this, which due to limitation of time, but also limitation of IP sensitivity issue, I cannot really show the result. But what we did afterwards is, of course, if you remember that this experiment started with a 30-degree temperature and very long photo period. So it's only natural for us to consider, okay, do we really need that high temperature? So we extended our experiment, and we followed up with a more mild and more practical situation. And also, of course, we want to know whether do we need that much far red or not. So we also applied a far red gradient. And I hope that I can report those results also reasonably soon. And also, what's important for us is to understand, of course, yes, we have seen the phenotypes. We have seen that far red plays a role. And the good thing about lettuce is that the genetic or the molecular network which controls the flowering process of far red is also of lettuce is reasonably well described. A lot of genes has been already reported, and they bear reasonable similarities between with other crops like tomato and also Arabidopsis, in this case, and brassica crops. So we have a lot of knowledge about the molecular control of flowering process. So what we will do in the next phase is also to understand via which pathways, via which genes are these being, is far red regulating in this case? Is it via for example, phytochemical interaction factors? Is it via certain hormone or hormone responsive genes? Then if we know, then that's of course, naturally our next step. So these are also currently ongoing research, and yeah. So of course, this is not done by myself. So we have a large consortium here that sponsored this project. We have lighting companies, we have a substrate company, we have vertical farming companies, but also seed companies, and of course the university, the Dutch Research Foundation, and Foundation for Food and Agriculture Research. These all supported this research. And of course, I have my supervisors, so Professor Leo Miselis and Dr. Ab Huvling. They are both they're the supervisors for this project. And the data you saw just now are also how coming out of the support from Ilse and Kai, who both worked on this project as a MSc student and as a guest researcher. Yeah, so that is pretty much my presentation part for today, and I guess this is slightly shorter than usual talks, but also what I would like to do today is also to make sure that we also have enough room for a nice discussion, especially I think with this result, it's quite a bit of there's something interesting on both at the scientific level, but also at application level. So yeah, so that's my presentation, and thank you very much.
Well, thank you so much, Yongran. I really appreciate. It's always exciting to learn something new, particularly in this space, controlled environment agriculture. So yeah, thank you so much.